You are watching a video lesson in the lecture series of Botanypedia to help you learn about various concepts of biology. I am Dr. Pavan Kumar and this e-lecture is about a brief introduction to biostatistics. So what is included in this lecture? We will start with an overview of biostatistics followed by application and limitations, types of data, and in the last we will look at few statistical terms. This slide gives an overview of the biostatistics. So what is biostatistics? It is a branch of biology that deals with the collection, presentation, analysis and interpretation of biological data. The term statistics is derived from the Latin word status, which refers to the status of any information collected by an investigator. Adolphe Coulet used statistical methods for the first time to solve the problems of biology, medicine and sociology. Francis Galton is known as father of biostatistics and eugenics. Our present understanding of correlation between two variables is credited to him. If you want to learn more about correlation, you can check the description below to find out the link that will take you to the e-lecture pertaining to correlation. Another biostatistician, W.F.R. Weldon, coined the term biometry. Bio means biology, metry means measurements. These are some applications of biostatistics. It has been extensively used in the field of genetics. For example, statistical methods were used by Mendel for performing his experiments and deriving the major conclusions from his work. It is used in molecular biology to study the sequence information, that is, to perform the sequence analysis of DNA, RNA, and amino acid sequences. It is used in agriculture. For example, standard regression techniques are used for construction of yield tables. In forestry measurements, we use this field to measure various characteristics like, for example, length, area of canopy, volume and weight. In pharmacology, it is used to find out the action of a particular drug. It is also used for comparison of action of two different drugs and evaluation of relative potency of a new drug. There are some other applications, like for example, it is used for testing a hypothesis, making predictions, to study evolutionary relationships. There are some limitations of statistical methods as well. Statistical technique can better deal with the quantitative data than qualitative data, as the latter cannot be studied properly unless given a numerical shape. What is the difference between the quantitative and qualitative data? This will be explained in the next subsequent slides. Statistical laws are not exact laws because they are based upon the values which are coming from averages, such as mean, mode, and median. Statistical data can be treated as approximation and not as precise measurements. Investigators need to have expertise in statistical analysis to derive inferences, as poor expertise can lead to faulty conclusions. Now we will look at the different types of data. So based upon the source of their data, there are two types, primary and secondary data. Primary data is factual and original data collected by investigator by performing experiments. Secondary data is that data that is used from previously published sources. So those experiments are actually or were actually done by somebody else. Data collected is in real time in case of primary data, while on the other hand secondary data is from the experiment performed in the past. Primary data is expensive because experimentation involves setting up labs which require infrastructure, funding, and skilled researchers. Secondary data, on the other hand, is comparatively economical in nature. Primary data takes longer time because to perform an experiment, to get the numbers, crunching them, and performing statistical analysis will take time. On the other hand, secondary data takes relatively shorter time. Primary data is more accurate and reliable. Secondary data is relatively less accurate. Now, based upon the nature of data, there are two types, qualitative data and quantitative data. Observations that are not numerical but descriptive fall under the category of qualitative data. For quantitative data, are represented in numbers, and hence observations here are numerical. Qualitative data does not have magnitude of the characteristic. On the other hand, quantitative data have both frequency as well as magnitude. In case of qualitative data, frequency of the attribute varies. For example, suppose you went to a park and you wanted to count the number of plants bearing red versus white flowers. So out of 10 flowers that you picked up, 6 were red in color and 4 were white in color. So the frequency of the 6 will become 6 by 10, and of that of white will become 4 by 10. Suppose you move to another segment of the same park and you repeated the experiment. The frequency of the red versus white may vary as you change your location or your experimental sample size. So the frequency of the attribute here can vary. In case of quantitative data, both the frequency as well as the magnitude can vary. For example, if you measure the height of students in a class, so you can have numbers anywhere between a particular range, and there may be some students who have exactly the same height, so the magnitude as well as the frequency can both vary. In terms of qualitative data, the conclusions drawn from the qualitative data are expressed as ratios or percentages. The quantitation of data in case of quantitative data leads to numerical measurement, which are interpreted in a meaningful way. Example of a qualitative data can be flower color, that is red versus white, which is descriptive in nature. And the example of quantitative variable can be anything that can be measured in numbers, such as the height of students, height of trees, weight of individuals, etc. Now we will look at few statistical terms that are widely used in statistics. What is a variable? It is any characteristic, number or a quantity that can be measured or counted. What is a control variable? It is that kind of a variable, a factor that is held constant during an experiment. What is a dependent variable? It is that variable whose value depends upon some factor or parameter. Like for example, a variable being tested and measured in an experiment whose value is dependent on the other variable is defined as dependent variable. What is an independent variable? A variable that investigator changes or controls and is assumed to have a direct effect on the dependent variable. In my subsequent slide, I will explain what is the difference between these terms in a little more detail. We will learn about various types of data in greater detail in another lesson and limit ourselves here to understand these three types mentioned here along with an example in the next slide. So this is the example. An investigator wanted to see the effects of two concentrations of a phytohormone on the growth of a plant species. So what is a phytohormone? It is a kind of a chemical that we apply on the leaves, can apply on the leaves of a particular plant to see whether it has any effect on the growth or it doesn't have any effect. So what the investigator did, the investigator actually used two concentrations of that hormone, and he wanted to see whether is there any effect of applying this particular hormone on the leaves of the plant on its growth. So he selected three plants of similar growth stages for his experiments, and he applied a solvent on one plant having no phytohormone and solvent containing dissolved phytohormone at two different concentrations, that is low versus high dose, and recorded the increase in the height of the plants after 1 week. So these are some representations. So all the three plants are of same age. On the first plant, no hormone was applied. Only the solvent in which the hormone should be dissolved was applied. And on the other two, these are called treated plants because on these plants were treated with the hormone. So of which one was treated with the low concentration of the hormone and the other was treated with the high dose of the hormone. Subsequently, after 1 week, the response was recorded, and that was the change in the plant height. So the question here is, which of these variables here are control variables, dependent variables, and independent variables? So the first one, the control variable, because here there was no the plant was not treated with any phytohormone, so this was held constant. So that the comparison can be made. This is your control variable. What is an independent variable here? The concentration of the hormone used. There were two categories of the concentrations used, low versus high dose, but both the categories were controlled by the investigator. So if you remember from the previous slide, the independent variable is that variable whose values or the values are defined by the investigator. So it is controlled by the investigator. And since the plant growth was dependent upon the values of these concentrations, so it was dependent variable. So by changing the concentration of the hormone, there was a change in the plant height. High dose led to more increase in the plant growth. So it is a dependent variable. So I hope you like this lecture. So if you want to read further on this topic, you can refer to these 3 references. Thank you for watching this lecture. If you really like this e-lecture, please like, subscribe and share. For more information, please visit my website www.botanypedia.com. On this website, you can freely download a PDF version of this particular e-lecture. Thank you. Thank you for watching.